Dan, and thanks for everyone joining. It's quite interesting. So I think we have attendees from pretty much every continent. So some of you got up very early, and others going to bed soon. I'm going to talk a little bit about computational neurostimulation. It's going to be sort of fairly broad introduction. Some of it work we've been doing, but broadly just really talking about some of the concepts and ideas, and maybe give you a bit of food for thought. So the way we think about brain stimulation is, in essence, that we want to use it to study or modify behavior. And I would perhaps argue that that's really ultimately the only reason why you should use this. This obviously means studying or modifying behavior in healthy participants, but also in disease. And throughout the talk, I'm going to refer to different kinds of non-invasive brain stimulation in a fairly loose way, just to highlight for those who don't know, a lot of it will touch on TDCS, or alternating current stimulation, TACS, and some of it on TMS, which are probably the most widely used non-invasive brain stimulation techniques. So I think the main premise for using non-invasive brain stimulation is to understand and study behavior and modify it. But when we try to actually do that, we have a problem, and that is that generally we have very little power to predict what actually will happen to behavior if we apply a specific stimulation protocol. And by predicting, I mean not making a guess based on, say, previous papers or some good reasoning, but to have a specific prediction based on a mechanism and a formal process through which we can describe that response. Now, there are challenges and reasons why that is the case, and a lot of them are probably related. Some of them are listed here. In part, it's, I think, due to there being a lack of mechanistic understanding, but then there are a lot of methodological issues, and that comes with the complication and the complexity of the technique, different ways of using these techniques throughout different labs, and often also perhaps the problem that these techniques can be quite variable and that effect sizes can be small. So jointly they contribute to that problem that if we make a choice for this specific stimulation protocol, the prediction of what is actually going to happen is generally impossible. So we don't really have a good way of choosing a protocol because we know that that is likely to have a specific effect with a certain degree of confidence anyway. And so the question is sort of what can the field do, or what are ways to address this issue and deal with this problem? Because obviously we don't want to just permute this parameter space and apply something and hope something useful happens, but we want to really have informed decisions and informed rationales for picking a specific protocol, say in a given patient group, because we know that that has a specific effect ultimately on behavior. And so there are lots of models we could use, and I'm going to sort of talk a bit about that and generally advocate that models have an important role to play. And perhaps there are two key families of models that ideally we'd like to look at. And in the first instance, it's models that tell us something about the putative mechanism of non-invasive brain stimulation. Give a prediction about what might happen given a certain parameter set, given a certain protocol, how that actually affects and interacts with neural circuits that we often cannot directly measure or assess, certainly in humans, and so maybe get some ideas about the underlying physiology. and perhaps even be able to account for the disease-specific changes, and so how stimulation might impact when we apply it in a disease. So that's largely concerned with the physiology and the impact on physiology. But then it's perhaps an even more challenging problem, and that is to develop models that can predict or make a prediction about what happens when we apply non-invasive brain stimulation ultimately on behavior. And sort of throughout the talk, I'm going to argue that while that is a big challenge, it is also possible, and the field is rapidly developing in that direction. So models to choose from and category of models. Many of you will be familiar with current flow models that try to make a prediction about where actually we are stimulating and which regions are targeted given specific protocols, say a TDCS montage or TMS applied over a certain region with a specific TMS coil. There's a lot of development on that, and biophysical models to optimize the waveforms of TMSA or how we should work on how these neural networks we are interested in, so on what happens to the computations carried out in these networks, and then perhaps even more challenging and therefore less when we change these computations. I'm going to sort of browse through three sort of topics basically, and one is about models that address the targeting. So current flow models, for example, models that help us define what we're stimulating, how much we're stimulating, and so on and so forth. And then models that tell us something about the physiology, say that model the morphology of cells or entire networks, and then models that tell us something about the likely behavioral change and what underpins it. So when we think about targeting, then it's quite interesting to observe that we generally don't know where we apply the current to and how much of it. That's particularly the case with TDCS. The picture on the right depicts that, where we generally have a fixed stimulator output. We apply that to everybody. We stick electrodes on the head, and we hope that that is eliciting comparable effects, but we don't generally know if we deliver the same amount of current into the brain and don't really know where the current is going to. And it's quite striking that that isn't even that often discussed. And early on, so very early work, more than 50 years ago, I think now, for example, by Rush and Driscoll, already made very clear that the amount of current entering the brain is of great consequence, and that isn't really that surprising, I think. But generally, we don't really pay much attention to how much current enters the brain and where to. So current flow models, for example, would in principle allow us to get an estimate about where the current goes and potentially how much of it, and we could use that in a variety of ways. To individualize of brain stimulation, on the left you see how we can use MRI images to segment the different parts of the brain, the different tissue types and structures, and derive from that a model how current would travel through these, if at all. And then in an individual brain, in an individual scan derived from such an MRI scan, we can create predictions about where the current will likely be and how much of it, or how strong the current is. So on the right-hand side, you see that for a variety of different electrode montages for TDCS. And it's become immediately apparent that depending on the montage, you get a very different distribution. It's also immediately apparent that the distribution is fairly widespread and that you don't necessarily get current at the location you might assume, for example, under the electrode. But used in this way, we can individualize the application. We can say, well, I want to apply in this subject my electrode such that a certain amount of current is delivered to a cortical region that I'm interested in. going one step further, we can use these kind of models, again, perhaps in individuals initially, to generate some new predictions about what type of montage may have a certain physiological effect. So this is work done in the lab a while back in collaboration with Maren Bixen and John Rothwell. And here it was really the idea to say, well, if we deliver current in a certain way to the target area in primary motor cortex, then perhaps because we have a slightly more uniform delivery of current, that has a stronger physiological effect than another type of montage that does not have that. And I won't go into too much detail, just to make the point that based on these kind of current flow models, we may make a prediction of what kind of effect should be occurring. And in motor cortex, of course, we can quantify it using TMS. So we would then apply TMS subsequently. And in this case, what seems interesting is that indeed there's a differential effect depending on how you specifically deliver current to the primary motor cortex. So it's, I think, an example how these models can allow us to formulate new ideas on what we may target and how we target it, and then to test those. We go from the individualized application of current flow models. We can look at entire populations, be it in patients or healthy subjects, and ask questions about population is really generally delivered to where I would think it is. This is an example from Karis Evans from a recent study looking at 50 subjects and 2 different types of stimulation. But the main point is here that once you start to do that, you can use these models to create probabilistic maps of where current is occurring and how consistently that current is delivered in your population to a specific location. So again, it's an interesting sort of development. Obviously, one could go further and use these sort of population-wise models to say, well, even though in a given subject I might not always have an MRI scan, I'm not so sure what's going to happen, I can make a probabilistic estimate that a certain region will indicate. Now, these tests, and I think this is often falling by the to emphasize, is that they do not tell us the effect stimulation. They tell us about the current and about the strength, but they do not tell us specifically whether we effectively target new tissue, whether we have the desired effect in terms of what we've said, and so on and so forth. Not tell. We also use human neuroimaging.
Hi, Sven. Sorry to interrupt. There's just been a few audio issues, I think, where you've started dropping out a little bit, so we've maybe missed the majority of this slide, unfortunately.
Okay. Can you hear me?
Sure. I can hear you quite clearly now. Yeah, I can.
Okay, so I'll just go through this again.
Great, thanks.
So the important point I was making is that all these type of models, or for TMS, models that try to predict, again, where the effective stimulation does occur, don't really give us an idea about the effect of stimulation, neither on the physiology nor on the resultant behavior. They don't really tell us what to expect, say, in terms of the change we can observe in a neuroimaging measurement, in a neurophysiological measurement, or behavior, as I said. So that's a very important limitation to bear in mind. They don't, therefore, really tell us if a target is engaged, and by engaged, I mean whether the stimulation is effective. And certainly, they don't tell us how it's engaged. And for that, therefore, we need to turn to other type of models. So we can really get a good idea using these models of whether we are delivering current to where we want to. But then what happens is not really addressed here. And that's sort of an important point that needs a different class of models. And those kind of models can tell us something about the physiological effects. So if we indeed deliver current to a specific area, a network, and so on and so forth, how do the cells in this area or in this network react, and what is the sort of overall consequence? Of course, we can measure that empirically, and there's lots of elegant work, a lot of it covered in recent seminars in this series, using imaging, for example, in which we can quantify the physiological effect, but then knowing why those effects arise is often very difficult. So that's why we want to perhaps create a model in which we can specifically sort of interrogate how the effect is occurring that we see at a large scale in humans. And again, there is work. These models vary a lot in terms of their focus, in terms of their level of observation. They go and focus on specific single cells, on entire neural networks, and anything in between. And I'm just going to give a sort of few cherry-picked examples of the kind of questions that one can address. The first point that common to all these models is that ultimately they can help revise what we think about the mechanisms of TMS or TDCS. But this is actually an interesting example from a study by Rahman a few years ago, who really compelling, interesting study measuring the fields on different type of cells in different layers, and then also developing a model to interrogate this further. And what this clearly shows is that for TDCS, this sort of idea that there's therefore pure depolarization or hyperpolarization it's not really true, even at the level of an individual neuron, because current has to go in and have the problem that either the soma is depolarized and the apical dendrites are hyperpolarized, or vice versa. And because we always need an anode and a cathode, both effects will occur in the brain. So this sort of whole idea about an unodal stimulation, certainly at the level of an individual neuron, it becomes at least something that people have started to think about quite deeply following those results and building on earlier work. These models can be very sophisticated at an individual neuron level, and I think probably owing to the development in computing power and others, really people have to use these very detailed neural models and embed them in entire that encompass the economy, as well as very fine-grained information about the different layers in cortex and the morphology. Ultra-realistic modeling of cortical circuits. And this example, which is by Angel Pityshev's group and first author Sabera, and you may have heard about this yesterday in his talk. These ultra-realistic models allow to really ask questions about where specific types of stimulation, in this case TMS, is actually targeting. And on the right-hand side, without going into much detail, you can immediately see that the local morphology, so the gyral folding, has a profound effect on the type of stimulation that occurs and the threshold needed to effectively target neurons in the different layers that were modeled here. So it really is, I think, an interesting step, because it starts to incorporate the local, but also at a mesoscopic, a microscopic level. I think some of the ideas on how we can use these models and learn from them go back quite a while. So one, I think, of the sort of seminal papers that maybe sometimes is a bit forgotten. 2005 already, but a very sophisticated cortico-thalamic model to simulate the impact of TMS essentially on these circuits. And what's interesting here was that the model was able to faithfully replicate the occurrence of the direct and indirect waves that we know can be measured epidurally when we stimulate primary motor cortex. And of course, these authors may have put a lot of effort in getting that to work, but a real advantage here is that once that model works, so to speak, one can then go into the model and tweak it and perturb it. So elegantly, I think in this study, the authors depleted the model or added different types of neurotransmitters. So you see, therefore, what happens or should happen when you increase GABAergic neurotransmission or when you mess around with the AMPA synaptic conductances, as seen here. And quite interestingly is, again, that then can be compared to empirical data. We can't obviously necessarily do that as easily, but in fact, there are quite a lot of studies that have given participants different type of drugs and then measured how the consequence of stimulation changed. And that way it can be used to develop new hypotheses of an intervention and then use experimentation to arbitrate between these. So it starts to go towards this idea that we want to link models and still a physiological response, but nevertheless. So this is another example by Molay Adekani and Fabrice Wedling and others who took, again, a quite sophisticated model of the circuitry, the sensory cortex, and essentially simulated the effect of TDCS in this network. When the network receives input, which resembles or is there to mimic the sensory input that usually reaches this area in a life, the TDCS as an extra transmembrane current, and then essentially studied how the behavior of this network changes under different types of stimulation. So when the network receives input, it generates so-called evoked potential, and this is shown in the middle here as stimulated evoked potential. And then one can add the transmembrane current and therefore ask how that changes the behavior. You can see immediately that when we increase or decrease the transmembrane current, which is reminiscent of what we often call cathodal or annual stimulation, then the SEP increases or decreases. And what was nice in this study is that the authors then looked at sensory potentials in rabbits that were recorded from sensory cortex and which they were able to, again, apply to different types of stimulations, so-called anodal and cathodal stimulation. And those sensory evoked potentials and the response to stimulation very closely what these authors found in the model. Again, whether that means that that's exactly how the brain works is a different matter, but it's a nice example of where the model now can be compared to physiology and be used to develop hypotheses, perhaps, of how these circuits respond to TDCS and test new ideas as well. So these were just a few examples. There are some more, and I think it's a rapidly developing field. But in a way that still doesn't get quite to the core of the problem, because we don't know if a certain change in the physiology then actually leads to specific behavioral response that we would like to achieve or like to see. And we don't really know why that would be the case. So what is the mapping from, say, a change in a transmembrane current onto an increase in motor learning or a reduction in working memory capacity. Generally, what we rely on is making a good guess, perhaps develop heuristics, and go back and look at the literature, hoping that there's some consistent finding, but we don't really have a good mapping that would allow us to say, well, given a physiological effect, this is what changes in the operation of the neurocircuit, and that ought to have a certain effect onto behavior. We have so many different levels of study the effects of brain stimulation and actually brain function more widely, that it is often very difficult to understand how, say, a microscopic change elicited by TDCS on, say, the membrane potentials in a pyramidal neuron trickle through the entire circuit and ultimately change what we can observe at a macroscopic level using neuroimaging, for example, or TMS, and ultimately what that really does to the system to change the behavior. So a big challenge is to think about how these different levels of observation can be bridged and what we really need to understand or describe in our models in order to generate a prediction of behavior. This just putting it in a more sort of direct example, we have these very elegant works by Lynn Bindman from the 60s showing the basic effects of polarization on cortical firing rates. And I think this was work, among others, that has inspired the resurgent subjects. But knowing that in a in vitro recording firing rates can be elevated doesn't necessarily tell us whether that's a good thing for a healthy human participant or patient, nor what it ought to achieve in terms of the processing of a circuit. Similarly, in humans, we have the motor evoked potentials, which kind of are the hallmark measurement of the effects of stimulation, such as TDCS, because we can apply these readouts very easily. But I think fair to say what on earth a change in a motor evoked potential means is really not clear. So again, we have this readout, but it doesn't necessarily tell us what the underlying mechanism is, nor what we should expect in terms of behavioral change. So that's really the challenge where I think the models are developing, but that's really getting at the question of computational neurostimulation as we certainly see it at the moment. So this challenge is verbalized here and shown here. It's really about finding and developing plausible models at a sufficient level of detail so that we can generate physiological but also behavioral predictions of what stimulation does. And sufficient detail is very broad. I guess often what one sees in the literature is that it doesn't require to make a model or simulate single neurons in all the intricate detail, ion channels and whatnot, but generally is sufficiently achieved at a mesoscopic level of observation. And in fact, sometimes a behavioral computational model in itself could be sufficient to make a good prediction. But what it really allows us, if we have such a model, it allows us to make mechanistic statements, to explain the behavioral phenomena that we observe and that we're interested in by reference to an underlying physical or computational cause. So these models would allow us to say, and to ask and inspect what happens to a neural circuit, a neural network, and so on, and how does it cause the behavioral change. Most likely, whatever we find is going to be a bit simplistic, if not outright wrong. But by having this formalism of a model, we can generate different hypotheses quite easily by modifying the model. And we can then start to compare that with empirical data. And so certainly that's, I think, a huge appeal for these type of models. Now, it's not necessarily easy to build a model, but it's perhaps easier than generally is thought. So I'm just going to give a brief example, but also to say that if there are any positive sides of the recent pandemic, it is that things like sharing of data, sharing of code, sharing of models is accelerating. And there are a lot of very successful, very powerful and advanced models out there that everyone can use, and often without necessarily deep modeling experience. Interrogate the models and ask questions on the effect of stimulation. So that's just to say that one doesn't have to be a modeler to embark on this. There is an enormous computational literature and lots of groups working on these models, not necessarily being aware that they could also be interesting for non-invasive brain stimulation. So it's just to encourage everybody maybe to look around, given a certain question, and try and do this. But in principle, it could work like this. We have a model where, say, we stick in a set of pyramidal neurons and a set of inhibitory neurons. And again, how those work, there's a lot of software available already that is fairly easy to use. And then we define what kind of network we want. So in this case, you see there are two populations of pyramidal neurons, and they're reciprocally connected to one pool of inhibitory neurons. And that's, of course, just a very simplistic model of a cortical circuit, but it is a start. And we define some rules. We may want to say, well, if there's task input and I operationalize this as just an input to one of the two pyramidal populations, and that will increase their firing rates. And then there's maybe some background noise, as we often see in biological systems. And I can then start to basically simulate, given a certain input to population 1 or 2, how does the sort of dynamics of the network unfold. And that may generate something that looks like this. So here you see the two inputs, left and right. So given to the one and on the left-hand side and right-hand side pyramidal population. These inputs differ, which could reflect, say, a stimulus given to the left-hand side and the right-hand side, or anything, something like that. And then on the right-hand side, you see the development of neural firing rates in this network. So in this case, how the pyramidal populations behave. And you can immediately see, which isn't so surprising, that when the stimulus is switched on, because the purple stimulus is so much stronger, it drives the firing rates of the pyramidal neurons in that population. Because these neurons are interconnected with an inhibitory pool of neurons, the other population that doesn't receive such a strong input gets gradually suppressed. It's a fairly sort of simple model generating, if you like, a prediction about how neural firing rates ought to look like. But what we can do here is we can generate a behavior. We can say, well, if any of the firing rates exceed a certain threshold, which is arbitrary, but say 20 hertz in this case, then we say that network makes a decision. And we can define the time point at which that happens, and that gives us the decision time. So certainly we can generate something that is akin to a response or decision time and to something like accuracy. We can obviously simulate a case where the input differs. So here you now see the example on the top where both populations receive the same input more or less. And what you can then start to observe is that the dynamics in the network change profoundly, that the threshold we defined is reached at a much later stage, and therefore also the decision time occurs much later. And that's not surprising, and it would sort of be a proxy to saying, well, on the one hand, the organism had to make a decision that was fairly easy because the right-hand response was so much stronger than the left-hand input, sorry, not response, input. Make a decision very quickly because the input that it received was fairly even among the two options. Now, this is a very primitive way, but it's nevertheless a way how we can say, well, this model now produces a behavior, and we can generate trial after trial with different inputs and play around the state of the network and derive something like a decision time or accuracy. And that's relevant because that is something we can derive in human subjects as well. We can start to think about tasks that may have a similar idea, where a subject receives two different types of input and make a decision whether it's one or the other that is stronger, brighter, or coherent and whatnot. And we can then start to compare those and say, well, is my model sufficient to describe the behavior that I observe? Clearly, this network is not claiming in any way that is how the brain works or it's a faithful replication, but it may be sufficient. One way you could leverage that is to ask now about the effects of stimulation. Many of you might be aware there's quite some interest in the possible nonlinear effects of TDCS. And that just really means that if I double my intensity, maybe the effect isn't double, but it may behave in an unpredictable way. Maybe it's quadruples or it actually diminishes. There's quite a lot of examples now in the literature. Why that is really hard to find out. There's some, I would say, proposals in those papers, but it's really hard to get an idea without, I think, a model that encaptures the complexity and nonlinear dynamics that occur in the biological system. So the example here shows you the three different cases. In blue, the control, and then in red and green, the two different types of TDCS, hyperpolarizing and depolarizing, that we apply to the network. And you can immediately see that when the stimulus is switched on, so the yellow area, the firing rates vary profoundly. And that would impact at when the threshold is reached and therefore the decision time. And another thing that is interesting that maybe perhaps without looking at something like this model, one wouldn't necessarily think about is that there's something called a pre-stimulus bias. So before the stimulus is actually given to the network, firing rates are already different because the TDCS is switched on throughout. So once the stimulus is switched on, this sort of model starts from a different origin. And maybe that's actually what explains this divergent firing rate. When we look at a simple task in which a subject, in this case actually a virtual subject, would have to discriminate between left and right-hand stimulus, and we can say, well, how do different intensities of stimulation affect the accuracy and decision time? And we can do this parametrically, which isn't so easy perhaps in vivo. And this generates a prediction of the behavior given a certain intensity. One thing you can see here that in terms of decision time and accuracy, there is a differential effect between depolarizing and hyperpolarizing stimulation, which is what we generally expected and what is consistent with what most people think about. But it's quite interesting also to note that certainly in terms of the decision time, and it's also evident a little bit in the accuracy, the effect of stimulation really only works when the inputs are not too different. In other words, when things are very different, think about that in a task, if the trial is too easy, then no type of stimulation is going to make you make a mistake in this case. You're still going to be fast and you're still going to be accurate by and large. So this type of stimulation does not, is not able to perturb your behavior in a specific range of inputs to the model. What you can also see is that for example, on the top-hand slide, in terms of the change in threshold, there seems to be some nonlinearity. So at low stimulation intensities, there's certainly hardly any effect. It takes a certain amount of stimulation before any change occurs. And then certainly for hyperpolarizing stimulation, it seems that at very high intensities, that effect reverses, which illustrates a possible nonlinearity. Now, this is in simulated data, so it's just really to generate predictions. How would behavior look like? It's about why is it? Just very briefly, the point is that there's no straightforward explanation. There are at least 3 reasons in this particular study. It's the limit on the integration rate of the network. So if you like a floor and a ceiling effect, you can't push the system beyond a certain range. And because of the complexity of the model and the interactions between populations that are inhibitory and excitatory, there are nonlinear effects on the neural firing rates, even before the stimulus occurs. And then there is potentially an inhibitory effect on the pyramidal neurons, certainly with hyperpolarizing stimulation. In principle, those are ideas one could test in principle, in this case, obviously in an animal recording and not in humans. It's just to say that this is just one model which has a lot of assumptions and features. There are others, and it's just for example from which is a very different model and gives sort of additional hypotheses or explanations. Now, these authors concluded that the effect we see is really because we modulate the probability of presynaptic release, that we only really see an effect when there's a sufficient number of neurons that are modulated. And when that effect occurs, we don't know, and that again can contribute to the nonlinear effects. The way the model is very nice is that it makes layer-specific predictions as well about how, for example, GABAergic interneurons are affected and what happens to the long-term effects, which in their case rely on glutamatergic plasticity. And a lot of this isn't necessarily surprising, but having the model would now allow you to interrogate different manipulations and really plan your experiments very carefully. So what about the behavior then? So the last bit. When we did the models I mentioned first, then I said they kind of resemble, and they have very successfully been applied to explain behavior in perceptual decision-making. Say when we, as you see on the left-hand side here, have to make a decision about whether a stimulus is predominantly moving to the right or to the left. And this is a moving grating stimulus. It's a region in the dorsal prefrontal cortex that has long been applied in this type of decision-making, and we can try to target that using TDCS, and therefore can try and polarize that region and ask whether subjects' behavior changes. And we can do the same now in our network, polarize the network in a specific way and see if the behavior of the network changes in a similar way. So this is the output, as I showed you before, that these networks generate. You see on the different trials, the inputs vary, and that would correspond to there being more evidence. And you slow the evolving neurodynamics and indicate the decision to a certain threshold. And so over trials, you can start to sort of build up the behavior of the model when you apply stimulation. So exciting, and you compare the model and the behavioral subjects in terms of the decision and the response time, they match really well. So the shaded lines are the observed, and the shaded lines are the model data, and the red lines are the observed, the solid lines are the data. And you can see that depolarizing stimulation decreases response times, vice versa for hyperpolarizing stimulation. But again, you also see that this only occurs for no-brainer, for difficult trials. When the coherence of the stimulus is large, subjects get it right, no matter what type of stimulation you apply. It gives some reassurance that the model is sufficient to describe this process. It doesn't claim that this is how it works, but it is sufficient, and within that, we can ask specific questions. Back to that pre-stimulus bias, what you can see when you apply the model, when you look at the model and different types of stimulation, that there is a residual effect on neural activity from trial to trial. So if you take the activity just before the stimulus, you can see that in some cases, the two pyramidal populations are different. And that's a carryover from the previous trial. And it looks as if that bias is actually exacerbated with depolarizing stimulation. And it's diminished when we apply hyperpolarizing stimulation. And we can now ask, for example, what happens to this pre-stimulus bias when subjects repeat their choice, so they say left 2 times, or when they make a different choice. And the model tells us is that when we apply depolarizing stimulation, they should make that they should have a larger repetition bias. So they should make the same response more often, simply because that residual neural effect that lingers on after trial is larger, or is enlarged. And with that, you would make the prediction that then if there's a long intertrial interval, that effect in turn diminishes, because the longer we wait until we apply stimulation again, the more time there is for the stimulus to come back to, for the neuroactivity to come back to baseline. And that's indeed what we see in the model. And why that is interesting is that we can actually go back now to the behavior for a participant and look at this, something that we would perhaps not have done, and ask, well, do they show this repetition bias by the different types of stimulation? And if it is, then we would predict that it should diminish the longer the ITI. And interesting, that's how the data plays out. That indeed, you get a polarization-specific effect, that the decision bias is increased and decreased, vice versa, for depolarizing and hyperpolarizing stimulation. And then that is really dependent on the intertrial interval. So for longer intertrial intervals, shown here 5 seconds, the effect actually is almost disappearing. So this is just an example how the predictions in terms of the behavior and the neural data in the model can tell us something quite interesting about the behavior in the subject and make predictions. Of course, the next step would be to go back to the model and perhaps equip it with other features such as learning and so on and so forth, and test how that plays out. But it just illustrates how it is possible in principle to develop models that can explain the behavior, and they can do so because they're linked to a biological mechanism that causes a change. And with that, we can identify candidate physiological processes that explain how stimulation impacts on the brain and how that ideally impacts on behavior. And of course, ideally, what we'd like is to develop disease-specific models, because in many of the diseases we are interested in, we're interested in applying stimulation, profound changes to the network occur, either in terms of their neurotransmitters, or if you take stroke, in terms of their structure, and so on and so forth. So we don't really know the effect of stimulation is at all comparable to what we've learned from healthy individuals. Now that's perhaps the way off, but the paper by Wendlin's group that I briefly showed is a nice example in that direction, where the author is sort of interested in epilepsy, so trying to think how network changes would occur in epilepsy and stimulation would interact with them. So just to conclude and maybe wrap up, the the use of models, I think, is really important. It's a big challenge for the field going forward, especially how to bridge the models that across different levels of observation, how to link them with models that look at targeting, such as current flow models. The purpose of all of this is to modify behavior, and that must always be the sort of final goal, I would say, in terms of our experiments, but also the models that we develop. And it is increasingly becoming possible, and the entry threshold is becoming lower to do these kind of things. Thanks.
Excellent. Thank you very much for that, Sven. So as hopefully you still have a bit of time, and this is the time where we can give you some questions to follow up on that. So if anybody has any questions, they can type them into the Q&A box. So one that we did have in a bit earlier on in the talk, which I saved till the end, but you've kind of discussed it in that summary. I'll still bring it up anyway, though. So can we use computational neurostimulation methods and techniques to better understand and develop clinical applications of non-invasive brain stimulation, which I think you've said there. Maybe I can adapt it slightly for that person and just say, how far away do you think that might be in the grand scheme of things?
Well, I think the question has to be how good are the computational models for different diseases? So I think in cases like DBS and maybe epilepsy, there are very interesting models that describe the physiological impact, and also increasingly how that might impact on certain capacity of the network and so on and so forth. And as I said, there's a huge community working on various models for different diseases. And to the extent where these models are quite successful, we probably can use them to add our stimulation on top, provided we have a good idea how stimulation would affect that specific model. Maybe, as I said, it could be in terms of a change in membrane potential, or we just operationalize it as a change in firing rate. If the model is more abstract, then maybe a change in a connectivity matrix, and so on and so forth. So I think to the extent where these models are there, a lot of them probably lend themselves to interrogation with stimulation. And that's, therefore, I think perhaps in many classes of disorders people are interested in, it's not that far away if people would start to use these kind of models that are already out there.
And a related question to that then separately is talking about individualizing that to the specific subject or patient. And so is that a potential with those disease-based models as well? And what I assume it didn't say in the question, but would it require various imaging techniques as well to make that possible?
It's probably a bit further away. I mean, it depends whether you would have a good idea what specifically is wrong in that particular individual. So if we talk about the targeting side, then yes, I mean, we can probably use provide a scan. We can that's done quite a lot already. But in terms of I would say the actual computational model that tells us about the physiology and the behavior, it's a bit more difficult. We would have to incorporate the local morphology, a bit like what just is about to happen and is being done, say, by people like Angel Petershev. Those models, I suspect, aren't straightforward to use in terms of their computational demand, but I don't actually know. It's certainly possible now. And maybe there are cases where there's a very clear marker of the disease in an individual and a model that would incorporate that. And then you could actually try and develop an individualized application. I think it's perhaps a bit further away, certainly in terms of predicting what the behavior would be in that particular subject.
Excellent. Thank you for that. Thank you for those detailed answers and your view on it. Different approach to this next question. So could you back up a few levels of complexity and say something about predicting peripheral nerve stimulation at different electric field magnitudes and frequencies? What are the best models and methods for predicting the behavior of this far less complicated system?
Right. I'm not that familiar with the models that are out there. You'd think initially it's a lot simpler, but I think it entirely depends on whether there are models that try and model how a type of stimulation, say TMS, would affect the peripheral nerve and how that would in a way, I would think there are good models out there already, but I don't quite know. Yeah, that's probably the best I can say about that.
Okay, no problem. And then the next question. So thank you again for the very interesting presentation. The question regarding the current flow models that simply model E-field. How much effort has been placed in validating them in terms of their numerical accuracy and or relative distribution accuracy? In general, could you please comment on what a robust validation of models would look like?
All right. Well, I'm not really an expert on that side of things. I mean, it's true there has been quite a lot of work. I think we heard Maram Bixen on Monday hammering home that point again. So there have been a few studies using intracranial recordings in epileptic patients or in non-human primates, and they seem to show a relatively good correspondence. There's some other work, for example, by Dylan Edwards, using these models and then doing some validation with transcranial electrical stimulation and motor evoked potentials. And again, that seemed to work quite well. I mean, personally, I'm not too concerned of whether these models are accurate or not to a certain degree. I think what is relevant is that they tell us nothing about the effect this will have. So knowing that there's current somewhere makes it very, there's a certain temptation to then say this is almost like an activation where it's red, I ought to see the effect I want to have. And that's really not the case. So I think that people keep over improving them, so diminishing returns with regards to what you actually need. So certainly for TDCS, if you think about the size of the electrodes and the size of the area we're stimulating, how good does the model have to be and how detailed? So I think they do well. They probably differ depending what platform you use, but there isn't that much work on comparing them directly.
That's a good point, actually. The comparison between the different platforms, like you said, I haven't seen anything that does that. Could be interesting to see whether they compare.
I think Lucas Parra and Andy Ho have I think they have a paper, but it's not much. I mean, understandably, perhaps so. It strikes me as quite boring to do it, but also perhaps everyone has an investment in their own platform, and they don't want to do that comparison. I don't know, but I wouldn't be that surprised if a lot of these models agree fairly well, to be honest. But there certainly will be quantitative differences, but I don't know how large the qualitative differences will be, and also not how actually the model changes. At least one needs to define some kind of objective function. So at which stage then would it be really a better model? That again requires validation, I think. And that's not easy to do either. I mean, these patients, for example, are very difficult to get and so on and so forth.
Yeah. Okay, and thank you again for that as well. The last question I've got for now, I think, is, so given the relatively recent use of modeling in NIBS in particular, are there any past NIBS results that you would now question or think would be worthwhile revisiting?
I wouldn't necessarily say question. I think a lot of the work over the past 20 years has been very exciting. I think what often has happened, and it includes work we've done, is in terms of the interpretation or thinking about what the mechanism is, I think a lot of that work just wasn't able to get really to the bottom of it. So I touched on that when we look at a motor evoked potential, we actually recall we're in a muscle, we're not even in the brain. And we still don't fully understand how a TMS pulse elicits that motor evoked potential, never mind what exactly happens if we add something like TBCS on top. So perhaps a lot of the conclusions, are probably going to turn out to be quite simplistic. And if you look at some of these models, there was the model from our own work and the work from forgot the name of the first author that I showed. They clearly show that they're very complex nonlinear interactions affecting several neurotransmitters and neuron types. Under Petershev's work for TMS, physiological curvature of the cortex and so on and so forth. So I think, yeah, that perhaps a lot of the concepts needs to be slightly revised. And it certainly has started to be the case for the concept of anodal and cathodal stimulation, which is in a well-controlled setup like in vitro hippocampal slice. But when you then go to humans with these large electrodes and all the scalp in between and whatnot, it becomes basically a concept that is riddled with problems. And I think to some degree, these sort of current flow models, but also some of the physiological work have started to highlight that. I think it'd be interesting. I think it will eventually lead to a revision of a lot of the rationale. So when people say, I want to increase excitability, and I think that's a good thing, and it will make people read faster or jump higher and so on and so forth. So this sort of whole idea that there is actually a widespread increase in excitability, and that produces, for some miraculous reason, a beneficial behavior. I think that will be questioned. Not that can happen, but what is it that leads to that? I think that's what these models are really important for.
Yeah, excellent. Thank you very much again for that, and all of your willingness to delve into those questions and give your thoughts on it, and for the presentation as well. We really appreciate that. I think the delegates will have had a lot from it.
I mean, if people have other questions later on, I'm happy to, if they want to send an email, that's fine.